With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
The question is, which of the following events takes place during post-transcriptional modification in eukaryotes? So starting with post-transcriptional modification takes place in eukaryotes of hnRNA, and this process mainly includes three events. We'll discuss all of these one by one, starting with the first event, which is known as splicing. So splicing is the removal of introns from the primary transcript. It is the removal of introns from the primary transcript of hnRNA. It is heterogeneous RNA. And the ligation of exon to form a continuous sequence which specifies a functional polypeptide. So in the same process, exons are ligated again together after the removal of introns. So the presence of exon in a polypeptide refers the functional polypeptide. Coming to the second event, it is known as capping. So capping of eukaryotic mRNA refers to the addition of 7-methylguanosine. So it is the addition of 7-methylguanosine, and that also specifically to the 5' end of the mRNA. So it is to the 5' end in an unusual 5' triphosphate linkage. So here occurs an unusual, and mostly there is linkage between 5' and 3' ends, but this is a 5' triphosphate linkage that is formed in between them. So this was the second event. Now coming to the third event, which is referred to as the tailing. So during this event, what happens? It refers to the cleavage of 3' end. Cleavage of 3' end, and then the addition of 80 to 250 adenosine residues to create a polytail. So, and then there is an addition of 80 to 250 adenosine residues. That is A residues in order to create a polyadenosine, that is poly A tail. And this process which occurs here occurs in a template independent manner. Template independent. And why it is known as template independent? Because tailing occurs by the addition of adenylate residues at the 3' end by poly A polymerase enzyme. So in this case, the enzyme used is poly A polymerase enzyme. And this enzyme helps in the RNA synthesis without the use of template strand. So it does RNA synthesis without using the template strand. So this process is known as template independent, as there is no template used in this process because of this enzyme. So if we see all these three events and match with the options we have, the first option says exons are removed from primary transcript, but this is wrong, as during the process of splicing, there is a removal of introns from the primary transcript. Coming to the second option, it says 7-methyl guanosine cap is added at 3' end of the RNA transcript. But during capping, the addition of 7-methyl guanosine is to the 5' end and not the 3' end. The third option is addition of poly A segment at the 5' end of transcript. That is, it is talking about tailing, but in this case, there is addition of A residues to create a poly tail at the 3' end and not the 5' end. So this option is also wrong. Coming to the fourth option, we have in tailing, adenylated residues are added in a template independent manner. So this option is correct. As already explained, the tailing process occurs with the help of poly A polymerase enzyme, which is template independent. That means it does not use template in order to synthesize RNA. Hence, we have the final answer as among the following events which takes place during post-transcriptional modification in eukaryotes is in tailing, adenylated residues are added in a template-independent manner.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.